Hi, thank you for having me today. My name is Leslie Farland, and I'm an assistant professor in the Department of Epidemiology and Biostatistics at the University of Arizona. Today, I'm going to be presenting some of our ongoing research on how we can study biological markers of ovarian reserve. So we just heard a really nice presentation which outlined some of the adverse pregnancy outcomes that women firefighters may be at risk for. I wanted us to also think about reproductive health more holistically and think about reproductive factors beyond pregnancy, including infertility and menopause. And that's because there's a growing body of research that suggests that reproductive health can really be seen as this window into long-term health risk. So we know that infertility can be emotionally, physically, and economically burdensome for women. We know that pregnancies to women with infertility may be at increased risk of adverse outcomes. And we also know that women who experience infertility may be at increased risk of cancer and other chronic diseases later in life. Infertility is also related to timing of menopause, and specific infertility diagnoses may be associated with premature menopause. Early age at menopause, we know from prior research, has been associated with greater risk of cardiometabolic disease onset, including cardiovascular disease, as well as stroke. So what do we know about these reproductive health factors among firefighters? Well, the answer is we know very little, but prior research has suggested that female firefighters were more likely to use fertility treatment than what we would expect in a normal population, the average population. And there's also increasing concern about timing of menopause and early age at menopause in the fire service. So our research group has been interested in learning more about an emerging biomarker of ovarian reserve called anti-Mullerian hormone. So anti-Mullerian hormone is secreted by the granulosa cells of the follicle, and it's used clinically, oftentimes in fertility clinics or obstetrics and gynecology offices, as a marker of ovarian reserve. So we see that AMH decreases as a woman ages, and therefore is associated with timing of menopause, premature ovarian failure, as well as it can be predictive of how responsive a woman is to fertility treatments. There's also increasing evidence that AMH may be influenced by environmental exposures. So AMH may have important utility as a biological marker of exposures that specifically target the ovary. So previous research has suggested that current smokers had a 44 to 56% lower AMH level compared to non-smokers. Research has also suggested that biomass fuel burning or fire burning in the home was associated with a 36 to 45% lower AMH value. And similar combustion byproducts to smoking and wood fire burning, including polycyclic aromatic hydrocarbons, are created. So in mouse models, they've shown that a certain type of PAH, as well as PM2.5, are associated with lower AMH levels. But little is known about how other respiratory or environmental exposures affect AMH in humans, and very little is known about wildfire smoke, environmental smoke, and secondhand smoke. Preliminary research has indicated this was conducted among Tucson firefighters who are less than 50 years old, and we only had 25 in the sample, so this is a relatively small sample. But our preliminary work indicated that incumbent firefighters had lower AMH values, a mean difference of about 0.61 nanograms per milliliter compared to recruits, which is equivalent to nearly a 6-year difference in ovulatory age. And when we restricted to among incumbent firefighters, each additional year in the fire service was associated with a 0.25 nanogram per milliliter decrease in AMH, which is equivalent to about a 2-year decrease in natural ovulatory aging. But as I mentioned, this is a relatively small sample size, and we were unable to adjust for important covariates of interest. So we're currently working on a pilot project, which has been funded by the Southwest Environmental Health Sciences Center here at the University of Arizona, which is funded by the National Institutes of Environmental Health, to measure AMH levels among a group of female firefighters. And specifically, we want to investigate whether AMH levels are lower in the firefighting women compared to age-matched controls. We also want to investigate whether there is a dose-dependent relationship between the fire exposure and AMH levels. And so in order to do this, we will be sending a recruitment email to an existing cohort of female firefighters, which we've probably heard a little bit about today, the Health and Wellness of Women Firefighters study. And we're going to be recruiting participants who are premenopausal and who are currently menstruating. We will be sending them a questionnaire, which asks information about their reproductive history, as well as exposures within the last year, specifically firefighting exposures, reproductive health history, demographics, and contraceptive use. And then we will also be recruiting age-matched healthy comparisons through kind of a friend recruitment process. And recruitment should be happening later this month, which is very exciting. In addition to the questionnaire, we will be sending participants collection kits to sample dry blood spots of the participants. And we know that AMH can be validly measured via dried blood spot. It's less burdensome to participants than doing a venous blood draw. We also know that AMH is stable over the menstrual cycle, so we don't have to time these blood measurements. And the goals of this pilot project are specifically to be able to compare the AMH levels between female firefighters and comparison women. As we mentioned, to also investigate this idea of a dose-dependent relationship between years firefighting and AMH levels. And lastly, I think from a research design perspective, we're excited to think about and show the feasibility for collecting AMH from dried blood spots on a population level among a large number of individuals. We're also interested in showing the feasibility for recruiting a friend comparison population and what that will look like among this group. I wanted to highlight some additional work that our research group is doing here in Arizona. So we'll also be measuring markers of ovarian reserve in a project led by Dr. Jeff Burgess, who will be speaking after me, for the Women Firefighters study, stress, cancer risk, and reproductive toxicity. And the purpose of this study, which was recently funded, is to evaluate the causes of stress, cancer, and adverse reproductive health effects in women firefighters and plan effective interventions to mitigate these conditions. So this project will be building on the firefighter cancer cohort study. We will be enrolling 100 female new recruits and 100 incumbent career and volunteer firefighters. We'll be assessing self-reported markers of stress, cancer, reproductive health, and occupational exposures. And then blood samples will also be collected to assess epigenetic changes as well as AMH changes at enrollment, as well as, again, 18 to 24 months after recruitment. So we are very excited to see the results of the pilot project, which is the project that I mentioned first, as well as the results of this newly funded grant. So thank you for your attention today. I would like to thank my collaborators and the organizers of this event, as well as our fire service partners. And I look forward to answering questions a little bit later on. Thanks, bye-bye.